Isn't it funny that we know so little about the very thing that enables us to know? How do we go from genes to proteins, cells, the electrical impulses of neural networks, the wrinkly fatty blob that is our brain, to how we think, behave, and see our world? I work on autism, which affects the social mind. Autism is one of the most biological psychiatric traits and is 50% heritable. But even in autism, where there is a known biological basis, we have no idea how we get from genes to how we interact with others. Autism is so complex that decades of work are yet to find a single biological marker. Biological markers and tests are important in autism because it's difficult to diagnose. Early diagnosis means early access to services, and these can really help a child to flourish. So how do we find these metaphorical needles in a multi-dimensional haystack? We've recently entered the omics era, which allows us to test lots of markers at once. Conceptually, if autism is so multifactorial, then we must cast our net really wide. I'm working with samples donated by families to the Australian Autism Biobank. I'm looking at millions of genetic markers, 800,000 epigenetic markers, and hundreds of gut bacterial species and metabolites. And that's for just one of up to 2,000 people. Good thing we have a souped-up computer. I want to understand how we go from molecules to the social mind in autism. So far, we've crunched the genetic data, finding that children on the spectrum are at higher genetic liability for autism. Our microbiome study is the biggest yet, and is offering some interesting clues into this controversial link between gut bacteria and autism. Our initial findings haven't found any direct links between autism and the gut, but there may be indirect effects from diet. And then we've also looked at the epigenetic data, which reflects the interplay between genes and the environment, adding data from the US and Denmark in one of the largest studies to date. Ultimately, we want to integrate these markers together and see if they can better predict autism when combined. How we go from molecules to the social mind is one of the last frontiers in human understanding. But to the parent whose child isn't quite like the others, the schoolgirl who gets by with a playbook of social cues, or the uncle whose talent for hyper-realistic art was overshadowed by his eccentricities, the quest for diagnosis is exhausting but incredibly validating. Finding these biomarkers will ease this diagnostic odyssey, which can help families thrive in a society in which we are all kinder and more understanding of how others see the world.